Good morning. We're going to start a new set of material this morning, starting to talk about, cell reproduction and inheritance. And we'll finish the semester actually with this topic heading, talking about mitosis, meiosis, and then genetics, both Mendelian genetics and just a little brief introduction into human genetics. The main topic now that we're switching from metabolism, photosynthesis, and respiration, we're going to talk about in chapter 10 is mitosis as a type of cell reproduction. Okay. So we're referring to, in this chapter, primarily eukaryotes, organisms that have a membrane-bound nucleus with the DNA, the inheritable material inside. Mitosis is a type of cell reproduction that allows for various functions, which even include asexual reproduction in a lot of different organisms, in particular those near and dear to my heart called plants. In animals and also the human, mitosis is primarily used to grow, increase in size, used to repair damage, cuts, bruises, abrasions, and maintain cell number, replace old worn out cells. But meiosis is a very specific type of cell division, if you will, to produce gametes in humans and other animals. And in plants, it produces a type of reproductive cell called a spore. So usually, mitosis occurs in cells that are diploid and produces diploid cells. Meiosis occurs in germ cells that are initially diploid, but at the end, when gametes are formed, those particular cells are considered haploid. And we'll talk about what those chromosome levels are in a few slides. So a haploid cell contains one set of chromosomes, typically from the parent that produces it. So the letter N is used to represent haploid. Gametes and spores are haploid, typically in animals and plants. Fertilization is the fusion of gametes, haploid cells, to restore the chromosome number or level of that organism. Oftentimes it's called diploid, or having two sets of chromosomes, one from each of the parents. So in the human, a haploid, or one set of chromosomes, that number is 23 different chromosomes. The diploid number is 46. So in the human, mitosis is used to grow from a single cell. We all start out as a single cell zygote. Mitosis occurs trillions, zillions of times to produce an adult. Within that adult, meiosis occurs within the genitalia to produce gametes. Okay. Before we specifically talk about these eukaryotic processes of cell reproduction, let's talk about prokaryotes. How do they divide? How do they exchange DNA? As you see with the bolded words, prokaryotes, bacteria, and archaeans do not have a true nucleus, so they do not have true sexual reproduction, that they do not necessarily form gametes. They divide asexually by a process that's referred to as fission. So do chloroplasts and mitochondria, that are organelles that we talked about in the chapter 8 and chapter 7 on photosynthesis and respiration. So, fission is a process in which the bacterial DNA, the bacterial chromosome, if you will make a copy of itself, and then the cell will split in half approximately at the region where the chromosome replicated itself. It's a very fast process. So some people call it binary fission, some people call it prokaryotic fission, some people just call it fission. So Fission means to split. So the splitting of one cell to form two in bacteria and archaeans is a very fast process. Conjugation is a means of exchanging a portion of DNA that's contained in a circular region called a plasmid. I don't see plasmid labeled, but in the donor cell in the figure, the plasmid DNA is color coded with red. So conjugation involves two strains of bacteria of the same strain. One is a little different than the other, so they're often referred to as strains. They fuse via a conjugation tube, or a pilus tube, if you will. One of the strains has this plasmid, again color-coded in red. The plasmid replicates itself, and the replicated copy migrates to the recipient cell. So you have the transformation of the recipient cell to be just like the donor cell by this migration of the plasmid DNA into the recipient. That is referred to as conjugation. When two strains of bacteria, think two strains of E. coli, they conjugate, one fuses with the other, and the donor cell injects plasmid DNA into the recipient, transforming the recipient cell. So that is considered to be somewhat a primitive means of sexual reproduction, if you will. So those are some means of reproduction and DNA exchange in prokaryotes. So the rest of the chapter is going to talk about chromosomes, eukaryotes, and mitosis within eukaryotic cells. So we need to understand what a chromosome is. A chromosome is a condensed portion of DNA wrapped tightly around histone proteins, visible because the cell is typically in a dividing stage or state. Chromosomes are highly condensed, and they are formed from long stringy strands of DNA called chromatin. A unit of histone protein and DNA wrapped around it is called a nucleosome. So nucleosomes are kind of considered to be like subunits of chromosomes, building blocks of chromosomes. The first step really in cell reproduction is the replication of the cell's chromosomes. And each chromosome will produce a replicate identical copy. This is referred to as DNA replication, and each strand of DNA will replicate itself to form a duplicate strand. Each of these strands is then referred to as a sister chromatid, and the chromosome that is then replicated or duplicated is often represented by something that looks like the letter X. The two chromatids or sister chromatids that form are most often still connected to each other by a small region that is referred to as a centromere. This slide shows a close-up from an electron microscope, a close-up photograph of a replicated chromosome. The centromere region is labeled there in the middle. So again, chromatin is, think of long stringy spaghetti. Think of a chromosome as being ziti. So think chromatin long and stringy, chromatin being chromosomes being short and condensed. Before a cell can divide, the first thing it does again is it replicates each chromosome. So then it can split those replicated chromosomes in half, and the daughter cells that form will contain a set of those chromosomes. The human, in its diploid number, has 46 chromosomes. That means 23 are inherited from the mom and 23 from dad. So you have 23 pairs of chromosomes. One of each of the pairs from each parent. 23 chromosomes from each parent. 23 plus 23 is 46. We refer to chromosomes that have equal size, shape, and genes that are found on them. We refer to them as homologous. So humans typically have 22 pairs of homologous chromosomes, and then the 23rd pair are referred to as sex chromosomes, either two X's in a female or an X and a Y in a male. So those are our 46 chromosomes that you find in our diploid cells, in all cells of our body. Haploid refers to the chromosome level that you find in gametes or spores in plants. I put the pea plant example in just to show you that the pea plant has a haploid number of 7. We have a haploid number of 23. A diploid number is when you consider the chromosome number present in a body cell or a somatic cell. We have 46. Pea plants have 14. Just double the haploid number and you get the diploid number. The cell cycle is referred to as the orderly sequence of events that a cell goes through as it divides. This particular slide shows you that there are 3 stages. Interphase is G1, S, and G2. Mitosis is division of the nucleus of that cell, and cytokinesis is division of the cytoplasm of that cell to produce 2 daughter cells. So cell reproduction starts with one cell. That cell replicates its DNA and organelles. Then the nucleus divides, and then the cytoplasm divides kind of simultaneously to form 2 daughter cells. The little phrase IPMAT is often used to try to remember the sequence of stages. So we're going to end this particular presentation talking about the longest stage of the cell cycle, which is referred to as interphase. G1, S, and G2. G1 refers to growth phase 1, when the cell duplicates its organelles and also cytoplasm, gets a little bit bigger. The S, or synthesis portion of interphase, is when each chromosome makes a duplicate copy of itself. That is referred to as DNA replication. Very important. Each chromosome makes a copy, and those copies are then called sister chromatids. G2 is when the cell prepares this system of protein fibers that are referred to as a spindle apparatus or a bipolar spindle. This spindle apparatus is, the proteins for it are produced, and the cell also goes through what is called DNA repair, and the duplicated chromosomes are checked, and any errors that might have been made are repaired. Then, once interphase is completed, and you notice how large of a portion of the cell cycle interphase is, some people estimate it to be 80 to 90% of the cell cycle. But once the interphase portion of the cell cycle is complete, then the nucleus and cytoplasm will begin division, and that will be our discussion material for part 2. So take care. I'll talk to you soon. Bye.